Hi, I'm David Yang, a PhD candidate at the University of California, Davis, and a tobacco-related disease research program predoctoral fellow. My research focuses on utilizing integrative approaches to understand the intersection of cellular signaling, immune response, and metabolic changes, and the role these compartments play in coordination to drive fibrotic disease. My current work investigates macrophages, an important immune cell type, and how they are changed by exposure to tobacco smoking and subsequently drive lung fibrosis. Lung fibrosis is a disease characterized by progressive scarring or thickening of the lung tissue. It mostly affects older individuals and afflicts millions worldwide. It's also a particularly devastating disease with a median survival of 2 to 5 years, which is worse than many cancers. There are many factors contributing to the disease, with tobacco smoking being a significant risk factor. But how this drives the actual disease isn't well understood. Our research has indicated that tobacco smoking may drive lung fibrosis through changing the characteristics of macrophages, an immune cell type thought to play a significant role in fibrosis. In the image on the left, we observe macrophages in the lungs of fibrosis patients who had previously smoked. These cells demonstrate profibrotic characteristics, which we can see indicated by the green cells on the image on the right. Delving further into how these changes occur with a variety of screening tools, we found that upon exposure to tobacco smoke, cells had increased levels of two activated proteins, AXL and MARX. We further uncovered that they were able to interact and form a complex. Furthermore, these proteins promote fibrotic signals in macrophage cells to potentially drive lung fibrosis. With these observations in mind, we looked at whether it would be possible to stop fibrotic signals in macrophages by targeting these proteins. Treatment with a peptide MPS demonstrated that indeed, if we do target this protein complex, we can reduce much of the profibrotic signals and changes such as VEGF and CD163 in levels in macrophage cells. To confirm our findings, we tested to see if this could be reproduced in an animal model of smoking-related lung fibrosis, where lung fibrosis was induced and mouse were exposed to tobacco smoke. We showed that indeed, smoke exposure, profibrotic macrophages are observed, and that the presence of these macrophages worsened fibrosis. With treatment, overall fibrosis was reduced as indicated by the reduction in collagen content in the lungs of treated mice. Confirming this, we see similar results with a reduction in fibrosis in another model where we directly inject cells isolated from pulmonary fibrosis patients with smoking history into the lungs of mice. In all, our work suggests that the AXL MARX is a novel smoke-responsive complex, lung macrophages, and may be a promising target in smoke-associated fibrotic lung disease. With that, thank you for your time. I look forward to any questions or comments.